The future of cancer research already exists. Most laboratories simply cannot afford to participate in it. Modern spatial biology is transforming how we understand cancer and the immune system. Platforms like Xenium, PhenoCycler, and CosMx are remarkable. But they require specialized hardware, specialized software, and major capital investment. For smaller research groups, teaching hospitals, and scientists in lower resource regions, that future can remain out of reach. Meanwhile, millions of ordinary brightfield IHC slides already exist. They are familiar, they are everywhere. But turning them into spatial analysis is painfully fragmented. A single analysis often requires segmentation software, registration tools, spreadsheets, spatial statistics, visualization, validation, and reporting. Every handoff wastes time. Every handoff adds complexity. Every handoff makes the work harder to reproduce. OASIS was built to close that gap. Not by replacing advanced imaging, not by replacing pathologists or biologists, but by bringing the entire brightfield IHC workflow into one accessible platform, running on the stains and scanners labs already have. What once required hours of moving between disconnected software becomes one guided, reproducible workflow. It starts with batch processing. Instead of analyzing slides one at a time, OASIS begins with the experiment itself. An entire data set is processed together, with consistent settings across every sample. A whole cohort of images becomes a single, reproducible pipeline, generating overlays, extracting measurements, and organizing structured outputs automatically. Manual repetition becomes automation. Disconnected steps become a workflow you can scale. Open any processed slide, and you can see the analysis behind every result. Cells are segmented. Marker-positive populations are quantified. And every measurement stays linked to the original image, so results can be inspected, not blindly trusted. For 2 markers across serial sections, OASIS guides the user through registration, checks the alignment, and only then measures how cell populations sit in space. When 2 stains come from the same physical section, OASIS can also support same-cell co-expression analysis, linking marker patterns back to individual cells. And here is what sets it apart. On a real tissue pair, a naive test reported a strong association, p equals 0.001. The calibrated test returned p equals 0.32 and flagged it as an artifact of shared tissue structure, not real biology. Same data. The software refused the false positive. That rigor is measured, not assumed. Segmentation is validated against expert annotations. Statistics are matched to an independent reference. Finally, OASIS produces reviewable overlays, summary tables, structured exports, and reports that can be shared, reproduced, and audited. OASIS does not replace pathologists. It does not replace biologists. It removes the software barriers standing between them and their science, making advanced spatial pathology more accessible, more reproducible, and far easier to perform using the pathology data researchers already have.